Hello researcher, lovable welcome to material research review. Today topic is two-dimensional and three-dimensional material application part 1. Nowadays using this material the researchers so many kind of application they doing like electrocatalyst and photocatalyst, sensor and electronic device, and biomedical field. Uh, the author this paper also they used the combination of 2D and 3D material combination. They synthesis like a graphene uh, graphene carbon nanotube web structure they prepared, and they did the application part like uh, brain tumor like a glioma infiltration. What is the highlight of this work? Like a new in vitro model to investigate the uh, malignant glioma infiltration. They did only today. We'll see this video. First of all, this paper is published advanced material end of 2018. Professor B. Tor in Italy. This article collaborate with 8 university like China University 5 and Italy University 2 and USA 1 university. Uh, Here the author synthesized the 3D interconnected graphene carbon nanotube web, and the application they did like glioma in infiltration in bioengineered. How they synthesize for this material? Already they published one paper in Nature Material 2011. They like graphene forms they synthesized. That procedure only for this paper also followed. Initially nickel foam commercially available. After that the nickel foam and methane, hydrogen and argon atmosphere, high temperature like more than 1000 degree they used the CVD method synthesis for the graphene nickel foam. After nickel attaching using the iron and HCl. So nickel once attached means it will form like graphene forms forming. So this first step they did. After second this nickel forms again argon, nitrogen and methane, high temperature. Here on the nickel sorry graphene foam side, it will form like a carbon nanotube. They said this structure is graphene carbon nanotube web. They say after the characterization for this material using SEM. Um, SEM you can see that clearly that nickel forms and deepen. They're saying like a few layer graphene, but in this case of graphene forms like a double layer only came. The comparison with the conductivity both and C and conductivity and elasticity, they used like IV curve they showing compare than graphene forms the graphene carbon nanotube web showing higher conductivity and good elasticity. Like going to application part, I am not familiar with biomedical field. In this article I am reading what I understand, that one only I will explain for here. Here the cortical culture staining, they taking confocal image. Here the nutrient stain with MAP2 and oxon with SMI 312. Both they used dye, and here the nucleus is the ostrich nucleus they used. So here without nucleus damaging they achieving the bright signal, that only you can see the image here. They compared with graphene forms sac fold and graphene carbon nanotube web, both they compared. You can see this figure, like wherever that graphene fold graphene forms, only that on the surface only it's showing, but in the case of graphene carbon nanotube web, it all direction is showing very beautiful material. And they took after staining, they took SEM image also. They observed on the that arrow mark they put, you can see the arrow mark from there, extend both action and dimension all directions, that CNT web, and also it around the twin side also it will growing. That image only they showing. They run the calcium experiment. Here they used indicate that fluoro 4 am. They compared with both 3D graphene carbon nanotube and graphene forms sac fold, both they compared. uh, Here the graphene sac fold showing the only that cyclotron side only it's showing the brightness, and for the 3D graphene carbon nanotube website is all direction is showing that image you can see. But other that theoretical calculation I cannot understand. So after the staining for the agrican and laminon protein, the extracellular matrix with the CNT web and graphene forms, both they compared. Here the experiment, for example, the fluorescent label experiment, U87 mg cell inside the CNT web grow. Here you can see the one they selected area they took, and different time point they did. Here 0 to 52 minutes they did. You can see 0 minutes to 52 minutes the cell migration, several micrometer migrating the cell. After that live cell imaging experiment with labeled like a fluorescent label U87 mg cell and cortical cell, both they mixed. Here you can see the cell migration, several micrometer, and that plot also relating is showing here. Here the glioma cell interfiltration interact with the brain experiment. Here they use like a 3D mapping of glioma infiltration. They use the CNT web, pre-blend Western and post-blend Western, with cortex and without cortex. You can see without cortex all direction migrating more, and with the cortex you can see very clearly migrating the cell with the X, Y and Z direction. After the compared with one plot, how it is the CNT web without cortex and with cortex, how it is the migrating, that plot they explained. What is the novelty of this work? That graphene forms on the surface, it will growing graphene, carbon nanotube 3D structure. That is the one novel for this material. And the application part, cortex network allow to study brain connectivity and neuronal dysfunction, And the glioma infiltration model, they explain very clearly in the 3D mapping. I also one question for this paper. Initially, they took SEM cross section for this material like 1.5 mm thickness, and after cultured, checked with the confocal image, cross section image. They said 635 micrometer. Both are not matching. Future possibility work, this material like 2D 3D combination, graphene carbon nanotube. Why not use the like silicon cyclotron use, and some plasmonic 3D material to insert. Maybe it's possible because plasmonic metal also light harvesting material, so it will useful for not only biomedical field, some other like a photocatalyst or sensor application, so it possible. So finally, thank you for your kind attention for watching video, and welcome to your valuable comment. Thank you.